So welcome everybody to the VOTU podcast. We're live on steamstar.net and we have a special guest with us today. So if you actually log into Steamstar Network, you'll actually see it's not just my face up there today. So we have Greg Simpson here today, and he is building on top of the EOS blockchain. And basically he's bringing your DNA the blockchain, or this is kind of the idea behind it. So welcome, Greg. Can you tell us a little bit about why you started this project and how you came up with the idea?
You bet. Thank you. Thank you so much for having me. It's my pleasure truly to be able to share a little bit about the project and where we're headed. I think it was inspired originally when I began reading some of the terms of use for some of the DNA sequencers, sequencing services out there. And essentially, when you read these terms of use, they gain ownership over your DNA. And I took strong objection to that. I felt that knowing what I know, I've been in, my deep background is mostly in computer science. I worked for Oracle for a decade and a half. It was one of their top troubleshooters when things got really bad and Oracle was being sued because some business was down. I'd get jetted off to wherever and have hours to get things back up and running again. So that was a great career, but it also got kind of boring. And I looked around as I started to age and wondered, you know, what could I do to really try to help the world? And I saw the advances in DNA were actually exceeding Moore's law. The cost to sequence somebody, the amount of information that was coming out about our genetics and what impacts they had on life, particular genes, whether they were on or off. And I just decided I needed to shift gears. I needed to do something different with my life. And it sort of flooded together. I mean, it's a little woo-woo to say, but it seems like the universe pulled, you know, suddenly there was sequence, get people sequenced real cheaply and easily. Stumbled on that, you know, whole idea of having safe data and keys and privacy and the whole sort of, I guess maybe libertarian's the right word, maybe anarchist is the right word, I don't know, but the government shouldn't have what we, you know, especially in our DNA, if there's anything that's ours, it's that. And thus the project, that was how it started, yeah.
So, okay, you saw that kind of holding a lot of information. I mean, VOTU stands for Voices of the Underground, right? So we're sort of very much against censorship. We're very much about, you know, talking about things that the powers that be may not want you to talk about, right? So,
well, I'm in the right
so that's
place.
Kind of, yeah, exactly. That's kind of why we're, you know, particularly interested in this kind of thing. And it says in your white paper that, yeah, they have so much information that they're actually withholding. And, you know, all of the advances that we could probably have by now if that information was more open, we're kind of being held back. Like there's probably a lot of...
Absolutely. Yeah. This is horrific. If you look at what genes they're not allowing us to know about, those typically trace directly back into big pharma money lines.
Yeah. Yeah.
I mean, it's right there. It's right there if you'll dig it up. And it's, you know, you can look in the database that the US government keeps about genetics and what we know about certain genes. And when they do research, they don't get to publish and say, hey, we've found the HNM5 that turns off AIDS. It can't, there are people that walk around, they can swim in a swimming pool of AIDS and they will never ever get it. And when it costs $350 to create the cure for one person, and now you want to distribute the cure, and the only cost is feeding the bacteria sugar to make more magic DNA medicine, it's ridiculous. Yeah. Why millions of people are dying, and the money lines inside big pharma is the killer. That's the killer. And yeah, I take objection. And I'm very excited that this project has maybe got a chance at stopping that, ending some suffering. That's really what we're about. That's really what we want to do.
Yeah, Okay, so one of the main purposes of this blockchain then is research of DNA, to find cures for certain things. And then the other thing, which kind of boggles my mind because I don't have much knowledge of this kind of thing, but editing your DNA. Somebody can actually edit their own DNA from the comfort of their home. Isn't that the idea? If they
That's
know
very
their own
true.
DNA, they can. Yeah, like that's for me. The only knowledge I have of genetic engineering is like what you do to our food and stuff. So when it comes to doing your own DNA, I don't really know how all that works. Like you showed, I think you have like a tool where you're supposed to be able to extract your own DNA, or I assume it's a sample of your DNA, rather than pull it. Because I imagine like talk about in the white paper about taking a piece of your DNA out. I picture like a computer just falling to pieces or the person falling to pieces like the way a computer would if you like deleted an essential component. You know what I mean?
Sure.
So like, can you explain a little bit of where I'm going completely wrong?
I got you. And
Yeah.
okay, so the science is DNA is in every cell, and it's copied inside every cell. And so if you take a few skin cells or a hair sample and you pull some live cells out of there, you've got the DNA inside that cell. Now, that's not going to, you've pulled hair out with your comb or before, and no harm, no foul, you survived. So it's not going to impact you when we take DNA from you and use this little guy, this USB device, to sequence it. So in 48 hours with this guy, if I took some of your saliva is the easiest way to get it. And we take your saliva, we put it in a little centrifuge, spin it around inside the centrifuge in the test tube. There's these little tiny microballs, and they've got holes in them. And the DNA gets caught up in there. We break the cell wall apart, and the DNA kind of spills out. And then it gets trapped in those little balls. And then we rinse it, and then we extract those little DNA segments from those balls, process it through the machine, and 48 hours later, we've got your DNA. Everything, every letter, all 6 billion letters that make you you. And we'll talk about that again. It's not really 6 billion for each of us because we share a lot in common. We're 99.9% the same. But it's those 3 million little differences that make you a redhead and me a blonde and somebody else a brunette. So the idea is we can very easily get it out. It's non-destructive, it doesn't hurt you. And we'll talk about how the project intends to store that. I know you want to get into editing, and yeah, I'd love to describe editing. I'm going to do it. We're going to talk about that, I hope. It's a little freaky. I mean, literally, it is. It's freaky. People go, oh my God, I'm a GNO. I've edited myself. I've changed my DNA. And what happens when you do that is you change a couple cells, and then over time, those cells make, one cell makes 2 cells, makes 4 cells, makes 8 cells, and your other cells die off. So that's how that edit happens. It happens over time. Now, the wonderful thing about CRISPR.
You edit it on the outside, and when you put it back in, it sort of replaces what's already in there. Is that it?
Yes, you got it. Over time. You don't change every cell in your body in 2 hours. It takes some time to propagate through your body. So then you're essentially somebody new. And there's lots of different ways to do this. You could take this CRISPR juice, and I'll describe it a little more in detail in a minute. But this CRISPR juice, you could basically throw it in a bucket when you get it back from AdGene. That's a nonprofit organization that would send you CRISPR for about $350. And you throw it in a little bucket, put some sugar water in there, let the bacteria, they ship it inside bacteria. The DNA editor is living inside this bacteria. So you breed more and more bacteria in that bucket. Now you got a whole bucket of DNA medicine. You could dump that in your tub and soak in it every day, just keep breeding more bacteria, and you would eventually, the propagation level is very low. Now, if you do something else, the other extreme is you would take this DNA juice, CRISPR juice, and you would add another chemical to it, which would help it get inside a cell, and then you could inject it. And once you've done that, it would be a little more efficacious. It would get to your body faster. So that's how all that works. Yeah, that's
sorry, Beans. I have a question there. Just wondering.
Oh yeah, sure.
Yeah, why does the new edited DNA? Why does it take control and take over rather than being swamped by the existing DNA that's already there?
Yeah, thanks for your question. It's a good one. Essentially, you're injecting over and over and over. It is a repeated dose. It's not one shot and done. You continually push the edit into your cell. And remember, as these cells divide, now you have two fresh ones that are going to replace two dead ones. So the old DNA in a bell curve sort of way dies off, and the new DNA comes in. So the edit has to be a repeated application. You wouldn't just sit in a bath for 5 minutes and get out and go, I'm edited, I'm changed. You'd have to keep pushing it in. Does that help explain it? I realized I didn't explain that very well the first time around.
Yeah, that's starting to make sense.
Not really how it's going to be done. You lie in a bath of DNA.
Well, you know,
just one example.
It's a super low way to do it. Okay, you're not going to modify much. You're not going to be very successful. There's other ways to do it. Which there's a thing called a gene gun. It looks like basically a pencil, and it's got a little spring in it. And you pack the CRISPR up in the top of the gun, and you put up little tiny bits of gold, because gold are not going to be rejected by your body. And you hold it against your arm and shoot it, and you blast the CRISPR into the cell. You just kind of speed it along and out. You could continuously do that. You could give yourself continuous updates of the edit. Yeah.
And is that kind of, that's probably like getting a tattoo or something. It's kind of like a...
Yeah, it's not comfortable. I don't like it, but yeah, it can be. It's not totally painful. That's actually for me an easier way to deal with it. Use the gene gun.
Yeah. Tattoo artists all say that they love the needle, that it doesn't cause them pain, and it's like therapy or some shit. So.
Whoa.
So some
Okay.
people apparently get used to it. Yeah.
I suppose so. Sure.
I'm curious. You don't have to answer. But, I'm curious what part of yourself or your own DNA changed, and I'm curious how long did it take then?
Well, so far I tattooed myself. I edited a piece of my DNA we call it non-encoding. Its other term might be junk DNA. What it means is the DNA doesn't create any proteins, so it doesn't impact any cells, and it's essentially dead space. Think of it, you used the analogy of a hard drive before. That's the blank space on the hard drive. It's really not doing anything in your body. So that stuff, the ROSA 26 gene is one we know very well. It doesn't do anything. It's totally dormant. So I wrote this DNA as mine and mine alone, and I used a translator on the web to take those words and turn it into CATGCC. It's a code, so I encoded it. Then I went to AdGene, had them build me that code, and I knocked it into my ROSA 26 gene. So the answer to how long, I don't know. I haven't run enough of my samples to say. I did run one to know that it's in there. I found it. I found my code in my new DNA. So I know it's in there. I don't know that every cell's got it or how far along it is in getting through my body, but that was sort of my FU to the powers that be, that this is my stuff and they need to stay out, you know?
So this idea with the blockchain then is that presumably it's all voluntary, right? If you want your DNA to, if you want, information on your DNA, if you want you to own your own DNA, you volunteer to put it on the blockchain. And I think I read that it works kind of, it's encrypted, so that basically it's like your keys, your private keys is your DNA, and your public keys is, what was it again? I forget now, maybe you can tell me.
No, that's fine. I can jump in. It's a little confusing. We had to work it out to where we could ensure privacy, and that's the most important thing, that somebody can't get a hold of that information and use it. The idea is, the problem with that is then the servers can't run bots or code that would look for patterns in the DNA that could stop, cure suffering, cure ills, improve conditions. let me really just segue just a little bit, if I could, on that topic, because it's not always about genetic defects. There's a gene called FOXMO3, FOXMO3, and that particular gene, when it's turned on, and genes have, you remember you got twisted pair, right? You got one from mom and one from dad, and there can be three conditions: both on, both off, and one on and one off. So what that means is people who have a certain trait or a certain risk for breast cancer, if they're both on, like a BRCA1, BRCA2 gene, that's a problem. That's why Angelina Jolie had her surgery to prevent it,
Right.
because she knew genetically she was at big time risk. And now, if they had been both off, she probably wouldn't have had to do that. She also could have CRISPR'd them off. I would have loved to have been in the room and say, hey, whoa, put the knife down. We can do it easier. We can do it better. And so anyway, this FOXMO3, if they're both on, that's the guy who's 95 years old, looks 30, he's got a case of scotch under one arm and a carton of cigarettes under the other, and he eats horribly, and he should be destroyed. He should look like hell, but he looks fantastic. Because the FOXMO3 gene, what we think it does is our cells die, and it's called apoptosis when they die and they sit there. They don't get cleaned out. They occupy space, but they're dead. Now, these wrinkles around my eyes are exactly from that. It's dead cells that haven't been replaced because the dead cell is still there. And I use the analogy, it's like the old lady that died in the apartment. Until somebody goes and gets the body out, there's nobody new that can move into that apartment. There's just no room, right? She doesn't want to live there with this. Okay, so what we think the FOXMO3 does is it goes in, it cleans the dead lady out of the apartment so you can get get the wrinkle replaced, get some cells in there so your skin tightens up again. And when we see this really clearly in liver disease and certain things like that these guys with FOXMO3, they can be full-on alcoholics and they just don't suffer from it. Their livers look as good as a kid's, Yeah. So, I mean, there's positives and well as negatives, right? Hodgkin's, we know one gene. Down syndrome, we know one gene. The list goes on. There's 10,000 of these things that we know are detrimental in our genes. But there's also some really big ones that are very positive and exciting. It's like, why not live a healthy life up till 90 and you don't have to worry about diabetes or weight loss or heart disease? These are all things there are answers to.
That could be amazing. I mean, that kind of sounds like we're going in the direction of building superhumans.
No, somebody's going to do it. Somebody's going to do it. I'm not saying I'm all down with it, but
I mean, it's already pretty advanced that we live till 90 now. So, I mean, do you think this kind of thing could lead to, I don't know, us evolving a little bit even farther, where we're, majority of people end up, if it became really widespread, if everybody had control of their own DNA and they could just take that, the DNA that helps them to live longer, do you think we could even go further than what we already are?
I mean, without getting too woo-woo, can I use that word in here?
Yeah, you can.
Without going too cliff on you. If you take the stories of the Bible literally, and Noah was 960 and Methuselah was 994, and then you wonder about the flood myth, right? Every culture on the planet's got a flood myth. So I find it very difficult to think that all the, two mosquitoes, a male and a female, flew into his ark and sat there while the toads didn't eat them. I just find it really kind of a stretch. But what I might buy into is that some of the stories about the Anunnaki are there and it's real. And they said, hey, Noah, you get on the boat with your wife, which the boat may have had a saucer sort of shape, and they got above the flood with all the DNA. If there were a civilization that was capable of collecting DNA around the planet, I buy the whole myth, the flood myth, a lot easier. Going, yeah, they could have gone out and collected a whole bunch of DNA from a bunch of different critters, stuck it on the ship, come back down. Now, what's scary or a little unnerving maybe is right after Noah, the average lifetime in the Bible is now down to 30 years. What happened there? What happened there? Maybe we're in a big test tube. Maybe you and I and the rest of planet Earth is sitting in a big test tube and some advanced civilization decided. Now, the reason they would do that is the same reason scientists use fruit flies. the lifespan of a fruit fly is like super short. So you genetically, if you monkey with a fruit fly, you turn around and can see the results of your experiment very quickly. You don't have to wait 90 years or 80 years. Just something to think about. I know it's a little woo, it's a woo for me, but yeah, I don't know. I don't know.
Yeah, you never know. Okay, so back to the blockchain and the importance of privacy and security. So, yeah, so what are even the risks involved? Like, if somebody got hold of your DNA, how can they use that against you?
Well, this is pretty interesting that here's how we intend to jumble the data to make it essentially impossible. We're going to take your DNA and we're going to pull out all the stuff we have in common. We don't need that. We don't need to store that because we know what's in common. We keep that as like the dictionary. And then we write down how you're different and how I'm different. And those are only 3 million letters, give or take. Okay, so it's only about 3 megabytes of data, not much data. Okay. So we can break that data into 60,000 pieces or 100,000 pieces and give each one a tag, a unique key, an ID, public ID. And that public ID might also say what gene it's from, that little bit of data. Now we scatter that, we intermix our DNA, right? We don't upload one me at once. We upload a bit of me, a bit of you, a bit of her, and a bit of her, you know, and that way it's all jumbled in there. No one can make any sense of it. And you generate the private key that then lists all your public keys. So you can reassemble your own DNA and say, this is me, but nobody else can without your private key.
Even though it's technically public, it's all jumbled up in there so that you can't actually make sense of it unless you have the private keys to pull it all together.
Exactly. Okay. And even scientists would have a difficult time using their forensic laboratories where, like, if you were a criminal or you'd been somehow, your DNA had been taken by the state, they would still have a very difficult time of finding you and reassembling you on that blockchain. And if they already had it, why would they go hunt for it on the blockchain? You know, they've got you, they know you. Well, here's where we could thwart that. You're editable, you're fluid, you're changeable. The STS is the STRs, sorry, the short tandem repeats that the forensic database, little tiny bits of DNA that you and your family would kind of share. And they can be changed. So if we change them, how are they going to use DNA to identify you? It's a brave new world in DNA, and things are changing quite rapidly. As a matter of fact, I just heard of a new technique for editing that came out since I wrote the white paper a month and a half ago that's even got cooler properties than what we were going to work with in Cas9. So yeah, it's going crazy. Now the blockchain, because it's spread out like that, because you hold the private keys, it's totally safe. But the benefit to us in leaving it open, you know, originally we thought, well, we'll just encrypt the file and that way it'll be safe. But we can't do any research on encrypted files. That server has to look at the data to go, okay, I see a pattern in the DNA. I see 20% of people have this gene on twice and 80% have it off. What's it do? Now the idea is we'll offer you an app where you can put in a separate file that's not attached to you, but is attached to your public keys, some health history. Hey, I have liver cancer. How bad? I'm dead from it. This is my mom's DNA. She died from it. Okay. So we know that set of genes may have something to do with liver cancer. Then when the numbers say so, because we've got 1 million people on the blockchain and we can do genetic analysis in math rather than test tubes, right? We're able to just look at the math and go, yeah, there's a pattern here. And I like to use the analogy for these guys that would mine DNA research rather than mine silly little answers to mathematical problems that throw away and waste energy. Let's do something good with those CPU cycles in that data space. Let's advance humanity, end some suffering. I mean, this is like what this project's about. And I think that for them to mine it, they have to be able to see it, and those patterns will evolve, and then we'll find out how much we don't know. I mean, we know a small bit right now. Even knowing 10,000 genes and what they do, we got a lot more to go, and we don't know what their effects are. But if you get the numbers all lined out, you can find the pattern, and then you ask the people, hey, are you healthy? No, I'm not. I'm really sick. Oh, okay. the really sick people are on this side, they're too turned on, and the really healthy people are too turned off. Well, let's everybody turn them on or off. I forget. Sorry, lost myself.
Oh, that's good. Okay, so yeah, I kind of like that analogy as well, the mining analogy. So it's kind of the people doing the research are the ones mining. Is that right?
Yeah, the miners, the hosters on the server would get paid in coins for issuing a report saying, hey, I found a new pattern that hasn't been found before in the DNA. And it's the reverse of how normal genetic research goes. You know, how it goes today is they go, some geneticist says, I'm going to go study colon cancer. So they go and get 500 people with colon cancer and 500 people without, and they take their DNA and they look at it. And the problem with that is you can't, it's very difficult to tell with that small of a sample what's just blue eyes versus brown eyes and what's really related to the cancer. But if you have a million, there'll be people with blue eyes and brown eyes on both sides of that equation, and that anomaly will fall off the data because it balances out, right? You've got enough of a sample that all the things that really aren't related to what you're studying, if you're drawing the line and saying sick guys here, well guys here, and we don't actually draw that line. We just look for a pattern. And when we see, oh, we got some offs and some ons, we report on it. We found, now let's go look at your health history and see, well, what do you have? What don't you have? And is there a pattern in the health history? You know, is your family no one in your family's ever had heart disease? No one. You don't have it. You eat like a horrible person and you don't have it. And then all the other people, well, okay, now we found a gene that has something to do with heart disease, and we even know what the combination is, 2 on, 2 off, or 1 on, 1 off, that is the healthy one. Now we can edit that in. That cures diseases. It doesn't treat. And going back to the whole reason the project started, that's this crap that they're allowed to get away with it. Because this is not hard. I'm not a rocket scientist. I'm a very smart guy, but I'm no rocket scientist. I just put together a lot of pieces that have been done by some really great people in terms of advances. The blockchain is one. The whole ability to sequence somebody for cheap is another. The fact that we know some of this DNA stuff, you know, what gene does what, is the third. And I'm just standing on their shoulders, putting it all together, and that's what this project's about.
So is this something anybody can become a part of then? Anybody who can research genetics, DNA, they can...
Yeah, anybody who can spit in a cup can play, okay? All you got to do is spit in a cup. And you know, we send you a little vial, you spit in it, you pour a little juice in there that preserves it so it'll stay usable for 6 months, viable, and then you send it to us. And we sequence it for you, and, well, for our customers, then we do the math to separate out what's you, fragment that data, put it on the blockchain, and if you sign up for our package, the way I envision it is we're going to take and buy you some EOS that you will then own, but it will be locked. And now how this works is kind of exciting to me because I didn't think it was possible, but I found out from the guys at EOS just recently that this is very doable. So you lock the coins, you hold the keys to the EOS, and you can lease them to someone else who wants to borrow your EOS to do some processing. It doesn't have to be DNA processing. It could be anything on the EOS network. So you lease those out on the market, and then when you say, you know, I haven't had a report in a little bit, maybe I should see if they've figured out anything new about my DNA. You go ahead and take your coins back off the lease. You dedicate them to the AGE project, and you let us borrow your tokens to run the processing on the servers and give you back a report. After you get your report, you put them back on lease, and you're making money. And the idea is we've tried to keep this cost as low as I could possibly do it. And the idea is, again, if we give you back that percentage, we'll make it even lower over time because you'll be getting paid back.
Right. Okay. So then there's a reason really to have the token is so that you can actually access your information as well.
Absolutely. And the token is what you use to we need a great deal of resources. Over 66%, sorry, almost 70% of our expense in delivering this service is disposable goods. It's consumables. It's stuff that we can only use one time and have to throw away. So that's a big fee. And if we figure if we do a token sale, we should have an idea of how many people would want to redeem and spend their money and get sequenced and avail themselves of our service. So if we get 100,000, we know how many we're going to have to buy of these consumables, and we can project these costs. We also know how many machines we're going to have to build or buy, so that we can service people. They don't have to wait 6 months to get their DNA results back. We can turn it. I'm planning on 30 days from the time the token sale ends. Sorry, we'll need a little bit of time to build the lab space out, because it's not going to fit in my basement. It's a nice lab, but it's really small. So the idea is we'll get some lab space, we'll get the machines in place, and then within a short amount of time, once we collect your spit, we'll give you the results back, and you'll be up on the blockchain secured. And, you know, I think everybody should have their DNA locked down, and the blockchain's perfect for that. The reason being is it's so easy to edit. You could literally be rubbed up against by some slimy guy and you get edited. I know, don't freak out, don't freak out. It's
yeah, it's kind of like, is that
if they were carrying CRISPR juice on their arm, yes, that could happen, but that's not going to happen. Don't worry about it. Don't worry about it.
Right.
Yeah. No,
in theory,
they're not spraying anything in the air. No. They're not spraying anything.
Okay, I'm going to open it up and see if anybody here has any questions. Daddy, you find this stuff interesting, don't you?
Hey, good evening, guys. Great project. Great project. I'm very, very impressed, especially now listening to you and see the utility of the token is absolutely brilliant. I'll definitely give my support. I actually read the white paper on 2 separate occasions, and I'm very impressed with the layout of the work, the way you explained it, and the actual use case is actually pretty huge. I think you probably are about to start something very special. I think there's use case in terms of cross-sectional studies against ethnicity. For example, I was looking at African American community compared probably to your Jewish community. Why the Jews are exposed to this sort of bloodline in terms of diseases, why they're mostly affected compared to African Americans, why they're probably affected to high blood pressure and sugar. So there's a lot of use cases, and especially I'm particularly interested in probably getting involved in a project like that. So I would want to know, how can I get involved? By the way, I'm the person who, I think I sent you some LinkedIn invitation, so I'm the person, so I definitely want to get involved in something like that.
Yes.
So that's something
I remember you.
want to get involved in.
I remember you. That's awesome. I would welcome any sort of help. You've read the white paper. I know we're LinkedIn buddies, so feel free to say, hey, you know, come up with something. You know, you probably know more about doing these projects like this than I do, and I'm really just dependent on a lot of people to give me good advice and say, hey, you know, you're going to have to do this. You're going to have to, you know, set things up this way so that it works out best. So I welcome any and all contributions. And what I've made with the rest of the team, the deal I've cut with them is we're just going to split, you know, whatever we get. We're just going to share it out. I don't care about money. I want the project to succeed because I want the suffering to stop, and I want the bad system, the bad players in that system to be unseated. They don't deserve that kind of power over us, and I think they need to be taken down. And now there's the tools to do it, so it needs to happen. The ethnicity question is very interesting. I tend not to see that as a genetic scientist. When I look at DNA, yes, there are genetic changes that have produced ethnic pockets or results. You look at Aborigines that have been like islanders down around New Zealand and things that are very isolated and lived in very isolated communities for a long time. Their genetic footprint is very different from ours. Now, it's not 6 billion letters different. That would make them a snake or something else, not a human. But they're very, very definitely genetic patterns, and you can look at it from an ethnic point of view. But I see the I see the data. I see the CATG. I don't see the Jewish person or the African person. But, you know, it's very easy to see that because, yeah, they grew up in a pocket. They are ancestrally related to each other. And the genetic changes that the sun, by the way, is the biggest genetic engineer on Earth. And every time you walk out in the sun, a little bit of radiation is actually editing your DNA. He does it all the time. And that's how evolution, when you look in plants and things especially, we can trace that to the amount of radiation they get. In other words, the taller trees in the forest are actually altering themselves faster than the shady ones. They're actually diversifying more because they're being exposed to radiation hacks of their DNA. Interesting, yeah?
There's one thing for sure that I would like to see, that you get a cross-section, a very broad cross-section of samples. Hopefully we can do a little bit of comparisons, especially for the people, because, you know, there's a big debate in genetics, basically what is genetics versus the environment. What role does the environment play? Is it just the environment or is it just genetics? And then we also have to consider the epigenetic changes happening over time. I know there have been some interesting studies in Europe, especially with people with when you look at a high blood pressure for say, and those who are isolated in specific communities. So I guess there's a lot of use cases. So as much as we're trying to find out what makes someone's more prone or less prone to a particular disease, probably it's more of a genetics rather than an environment. So there's a lot of use cases that probably can put.
So the question there from Sir Cork, so he says, what keeps CRISPR DNA drives from going viral? Species skipping and creating great havoc. The premise is to release a modifier drive which reverts the prior drive's effects, except it takes generations. Further, a smart high school kid can do this. What prevents Jurassic Park and mass DNA drive wars between opposing species, modification opinions? This is like heavy stuff.
That's a loaded question, and it comes down to nothing. That's the real answer, nothing. But I think every genetic engineer I'm aware of, anybody who looks deeply in this topic, we've sort of, they have sort of, and I've gone in with this as well, that you don't cross the germline. Now, what that means is that any edit is constrained to that being. If you don't cross the germline, you don't edit eggs and sperm, you don't pass it on. So the edit dies with you, and it's non-transferable to your children, right? So if you want to have, if you want to have,
devil's advocate.
I love that, dude. If you want to have, you know, hollow bones and wings, you could do that, but your kids wouldn't have hollow bones and wings unless you cross the germline. And we've sort of put a moratorium on that until we're understanding it. Now, they're willing to do it with mosquitoes. They're willing to do it with other species, but crossing the human germline has been kind of verboten. The truth of it is nothing stops the great havoc that you mentioned. It could occur.
I think I saw Ankak Bardi asked, would the DNA editing have helped Angelina Jolie if she had already had the breast cancer? In other words,
that's kind of it's a good question, and it's kind of a race at that point. Once someone's developed cancer, you can undo it. There are better ways to treat it at that stage, and what I think we could have done for someone who actually is already full-blown, you know, manifesting cancer is what they did for my favorite GMO, Emily, in the white paper. They took her T cells out of her blood and they edited them so that, I'm not going to be able to hold the talk. So imagine both my fists just bouncing off this, right? This is the cancer here, and I'm bouncing off of it because I can't recognize it. And that's her T cells attempting to kill her leukemia. that she had. And what they did is they took the T cells out of the body, they edited them and gave it a claw, and it now can recognize that cancer and actually destroy it. That's why she's cancer free. So you wouldn't necessarily go edit the BRCA1, BRCA2 that developed the cancer, although that would help for non-reoccurrence, right? It would definitely reduce her risk for reoccurring after, but to get the cancer out of the body, you have to go at it a different way. Does that make sense? Did I answer the question?
I think so. And Cap, does it mean like you could use it as part of the treatment, right? As
yeah,
part
sure,
of the
absolutely.
To avoid, yeah.
And it would be a long-term, it's more preventive than treatment. You know, it's setting your body up to not really get it. Yeah.
And when you've had cancer, usually it means that you're prone to having it again. So it's a good part to have. Yeah.
Absolutely. No, you'd want to edit her straight away.
Yeah. Who else have we? I saw earlier from was just Devin there, yeah. He wears factor 50, so son can't change his genes. Let's see. Crazy BG, do you want to say anything? Or are you just kind of like
I'm completely speechless at the moment. I've just been sitting here trying to take it all in. I suppose one question I do have, and it might be I don't know if this is maybe a right question to ask or not, because you've mentioned in the terms of cancer. So, how effective would this be, for example? In the future as a technology against things like, I don't know, like HIV?
Oh, yeah. The HNM5 gene, it's in the white paper, and there are people in Northern Europe who are completely immune to HIV. They could swim in a swimming pool of HIV and not get it. Their bodies have no receptors for the virus to grasp, and that's a genetic change from those people that can get it. So we know that if you've got that gene, that particular gene that we've identified, both flipped on, you're at risk for developing a receptor to HIV. If you flip them both off, your body doesn't build that receptor, so the virus has nothing to bind through. It's a lot like the T cell in the cancer. They just don't meet up. So the virus kind of slides through your body and goes away. And there's lots of diseases that we believe are very much tied to that sort of mechanism, that the DNA controls the protein that controls the cell that creates the receptor or doesn't create the receptor. And when you shut them all off, you know, I don't even know how deep that list goes. The idea is we need people on the block. Yeah.
Yeah, does this mean like there are people who have genes, DNA that is valuable to other people if they could, you know, basically get their hands on it?
Oh, absolutely. Well, we don't need our hands on the DNA per se. We need our hands on the code. And that's what the code looks like to turn off the AIDS receptor. So now I can go to AdGene and say, hey, I want this code knocked into my body. And AdGene will fabricate this with their 3D printers. They're printing a molecule at a time. They stick it in the virus, or the bacteria rather, they send it to you and you eat it. Well, you don't eat it, you shoot it up or swim in it.
So this doesn't just necessarily mean then that the coding is to fight something, but it can actually be used as a preventative?
Oh, absolutely. You would, I mean, look at those people that waltz through life and, you know, why do two people work in a toxic waste dump and one gets deathly ill from millions of cancers and just gets wiped out, and that other guy just nothing happens to him? And I believe for one, most geneticists believe for two, and most people will come to learn that there's a genetic change that makes this person more capable of not becoming susceptible to that gene. Oh, sorry, that disease or that sickness. Or, you know, the subject came up a second ago, whether it's environment or genes. Well, I'm sure they both play a part in just about everything we deal with. But our project is about isolating the advantages. What's the best DNA for me? And while it's mine, it's also mine to edit and say, hey, you know, I don't want to be susceptible to H1N1 viruses. I don't want to get the flu. Well, what do I have to do to not get it? And we'll learn that if there's enough people on the blockchain where we can actually get some data out of there that says that's the pattern. So yeah, we really don't, genetic editing is, someone just said it, it is more preventative than curative. You're not going to run around and, oh, I've developed this sickness, I need to hurry up and edit myself. It's almost, you don't want to approach it that way. You want to approach it that I want to set myself up with the best DNA to have the longest, healthiest, happiest life I can have. And once I have that, then it's up to me to eat okay and not swim in toxic waste pools of you know, horrible chemicals that are going to end up killing me. So there you go.
There's another question there from Sir Cork. Should we modify mosquitoes to eliminate malaria and then yellow fever, then hepatitis? And he goes on to say, are we going to be undoing what we have done forever? Sir Cork, why don't you just get on mic? He's going to hear this like in 2 minutes because he's not watching the live stream. But yeah, that's the question anyway. I think the rest is just,
yeah. That's already underway. There's already people, scientists working on that, the mosquito thing, and they're doing what they call a gene drive, which that means they've crossed that germ line, saying they've edited the sperm cells and egg cells of the mosquitoes so that they pass this attribute on to their children. And they're actually pushing that. It's happening. It's being done right now. They're able to make malaria-free mosquitoes that won't transmit malaria. And they're also playing very interesting games with, I find it a little risky. It's outside of the stuff that I'm into. But it's pretty crazy. They're able to predict now how long the population would be overcome if they introduce the GMO mosquitoes into an environment.
Yeah, so you're saying that we can edit mosquitoes to not have malaria, but can't they still pick up malaria and carry it, even if, like, depending on what country they're in, or?
Well, that's the idea. You make it so that the malaria virus bounces off the mosquito and it doesn't attach. And you know,
okay, so
you see the mechanism, right? Yeah. So now they don't carry it, and they may get malaria in them, but it won't be transmitted because it won't live. It won't have a way to attach.
Okay.
Yeah, it is risky. It's something I don't want to play in.
Yeah.
Yeah.
It's someone else's ball game. But it's exciting stuff. I mean, there's so many, you know, advancements that well, I don't even know how much it can do.
Yeah, I just wanted to add just a little bit of it. In terms of the genetic editing, there's actually a couple of good cases that are out that probably people who, if they're interested, they could actually have a read on. One famous case is the one called Jasinga. I'm not sure if I have his name correct, but anyway, that guy, he had one of those biochemical pathway. He was lacking one of the enzymes, and basically what the scientists did is that they basically they went in and they edited that DNA. He was cured for a period of time, but he later went to develop some allergic reaction and later died. So that is a good case that probably people, if you're interested in, you can actually read up on. It's actually quite fun.
Where can we find
Absolutely.
that?
Jasinga. You can also look at there's something known as Medscope, or you can also read it up on ScienceDaily.com, or you can get it at JAMA, or you can also get it at Science Journal. There's a couple very good articles, peer-reviewed articles on it. It's not that hard. Basically, put it like this: your DNA is almost like a normal code that someone will write on, let's say, on Steamy blockchain. And basically, when someone writes a code, there is certain processes that produce, particularly whether or not it's a protein or a carbohydrate, whatever. And what happens is that these viruses, in order for a virus to affect someone, there must be something that is known as an epitope, or something on the cell or on the organism that has to attach. If there is no attachment for that virus or that bacteria or whatever to land and touch, then someone is basically, they cannot be affected. Now, there are specific genes that when you code it, basically that particular region where that virus normally attaches, basically it becomes null or void. So it doesn't have any effect. So basically, you can go into the cell and you can, for example, HIV, you can target the specific places where the virus usually attaches. And if you can edit the DNA, and basically you can make that that virus basically null, because it doesn't have a place where it can attach and cause that sort of change of reaction, then it basically, the virus becomes sort of ineffective. But HIV is something that is special. You know, it's something that it constantly changes. But there are a group of people, as he was explaining earlier, that has that genetic what we'll call a mutation, where basically whatever the virus does, it basically does not affect them, because there is no part in that person's body where that virus basically can attach and then probably imprint and then basically change the cell around. And that's basically how you should look at it. It's like, it's a code. You can basically change a code by affecting what that particular virus really affects, basically.
That's exactly right, and well said. It's awesome to hear somebody explain something I talk about constantly, and because, oh yeah, I forgot to say that. It's beautiful. Thank you for that. Sir Cork, I wanted to say that, you know, one of the things you brought up in your chat is the ease to, given what you want to tell CRISPR to do, not the easy part. Yeah, that's the trick, and this whole secret to getting enough people on the chain that we understand what the DNA, what particular genes turn on, turn off in terms of proteins. That's just a numbers game. The more people, the better. As far as those guys, us becoming homogenous because we go, oh, that's the perfect DNA. I don't think that people are going to see that. I think we're going to find instead people coming out and going, I want to have leopard spots. You know, I want scales. I want, you know, look at the way kids put stuff in their face, and we get tattoos. We do things. They're very into their own.
There's going to be a lot of hair changing. You know,
right. I want purple hair for real.
Yeah, exactly. Yeah, that's going to be the biggest things. I'd say that's how it'll take off at first. Is just that's what people are like, superficial.
I do believe it's down the road, that it's not something that we're going to see in the next couple months, but it's faster than we think. That somebody's going to figure out, shit, I can get snake skin, you know, and I like it.
Yeah.
Right.
Well, it's one of those things where the second it's available, like, I think that's what people will jump on.
X-Men-ish.
You know?
I love that word, X-Men-ish. Yeah, you can go there. I mean, CRISPR will be a tool that can do it. There he is. You guys are great.
There was a comment from Kitty Girl as well. Her father had a genetic mutation in his lots of letters gene that caused the condition that led to blindness, kidney failure, anemia, and other problems. Battled it for years. His twin brother had it as well. Myself and other family members have to be tested for it periodically to see if it's manifesting in us, and I'm planning to write a post about it on SteamIt. Yeah, I suppose all of those things, if you can isolate them and figure out what the gene is, then you can basically avoid it.
Oh yeah, anything that's out there and labeled genetic disease or genetic contributed diseases, where there's a genetic component to the issue. We'll have it and we'll nail it. And again, there's only about 10,000 out of about 60,000 that are known. They're known encoders, that we know 10,000 genes and what they do, but we don't know the other 50,000 that we know encode proteins and encode cholesterol and create other things in the body. So they're active genetics, but we really don't know what they do yet. Now, were we to get 1 million people on the blockchain and set the computer loose and say, hey, go figure it out. It's a math problem to solve. Draw the lines, figure out, divide them into categories, and then go query those people and say, hey, are you a healthy person or a sick person? Well, if you're sick, what do you have? And then the pattern emerges that we understand what that gene is. It hyper accelerates genetic research. It takes genetic research and gives it a boost to the moon. And the answer is we'll know. And what we do with that knowledge, I don't know that I'm in a position to answer that.
Yeah.
Just not.
And if I should just add, just to answer Sir Cook, in terms of the research and what this project and of its intentions, what I'm seeing is that basically you don't really need to have a background in genetics to basically to join into a program like this, because what is really needed is we need more eyes, because the more people we have working on this, the more eyes we have. Sometimes something that I would miss, you'd be able to see it, and basically you can compare that to a wide cross section of samples, and you can say, you know what, this particular gene is common to 7 or 8 samples that we have. There is something about this specific gene here that has some sort of significance, and that's where you will use your statistics, and you can make whether or not that data is statistically significant, or whether or not you basically reject your particular hypothesis. So all that is needed is that basically a lot of eyes and a lot of people who just willing just to go there and just play around. You now have a blockchain where you can just go and look up a particular disease, look at a sample size, try to form an association, make an hypothesis. Okay, we're saying that CATT is associated with XXX. 7 people in a sample size had CATT. This is a clinical case. This is what happened. Okay, now I can basically conclude that out of my sample of 7 people, the statistical significance was 1.5. Therefore, I reject the hypothesis, and this is basically, and you just presented it, you write a neat write-up, and you let somebody else carry on the task. And that's basically how you problems are solved. Problems are not necessarily solved because of geneticists or people who work in it, but basically people like me and you and everyone else who can basically recognize those similar, very small things that we basically may ignore.
Sorry, Duddy, I was reading someone's funny joke. You are right. You are right, but I think we can even do better than that. My uncle's background is he worked at a laboratory, a government lab in Tennessee, and was the creator of what is machine learning, what has become machine learning. He's a very brilliant mathematician. And the idea behind our project is we're going to use some of his techniques to mine this data. Rather than putting a bunch of eyes on it, we're going to put a bunch of eyes on some very specific reports that say, hey, I have statistically found a pattern in the DNA. And we know that it's multiple genes involved. We know that it's multiple conditions of these genes. And that the idea there is they're not going to have to be 1 billion people studying this stuff and coming up with theories, and we're going to say anomaly, 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 anomaly on that gene. And that is what, and the healthy pool, the sick pool, or the positive pool, the negative pool, the advantaged pool and the disadvantaged pool, right? So the idea is we let the code find the problems for us, find the data for us, and bring it to us rather than having to go out and come up with well, I'm going to have to write a function to study the data this way or see if that's involved. The idea is let the data bubble up its own anomalies. I hope that makes sense to you.
I mean, it'll still like all blockchains. In order for it to really have value, it requires participation. So, so how do you think people are going to actually want to give up their DNA in order to be a part of this?
Well,
what do you think will?
Well, I think it's important to realize that they're really not giving it up. They're securing it for themselves.
Of course.
They're putting it on the blockchain where it's safe. And what I mean by that is because we know it's so easy to edit, okay? And I hate to say it, but nefarious players could be doing things without our knowledge. If you have a baseline of your DNA stored on the blockchain, it's nothing to go run a comparison and say, hey, have I been altered in the last 5 years, 2 years? Is there anything changing with me? And we know it changes. Everything changes. The sun changes it. You might be exposed to some toxins that might have done some genetic damage to you. So there's lots of, just the simple age processing is theorized that there's some damage there. We know it. We know that's probably true. So the idea is having a baseline on yourself protects you against nefarious players messing with your DNA, and it's yours. You should know it. You should own it. And I think, to get involved, we're talking a couple ether, a 2 ether is what I'm trying. It was at 605, I think, when I ran the numbers last. So the idea is, give us a couple ether, we'll get you on the blockchain, we'll get it secured, it'll be in your hands, and later down the line, when we grow to 50,000 users or 100,000 users, we can blow away all genetic research, just simply because we'll have the numbers, and we'll be able to learn so much so fast that it'll change the world.
And you were telling me yesterday as well that the cost of this is really coming down.
Absolutely. The price of this device, which there's a little inside it right now, is a white cell that goes in there, and it's got a little hole that you dribble the DNA down, and that price is falling fast. And as it becomes solid state, which is the end goal of the company that manufactures that device, then they're going to be really cheap, and that's going to allow us to get even more people access to the service.
Rhonda K wants DNA, a DNA expert? Oh, I see. I thought you were looking for DNA in order to be an expert. I got it wrong. But yeah, it looks like you have another another connection there. Question: do we need to worry about governmental interference with this project? Okay, girl.
Yeah, I'm scared of that. I'm not stopping.
You're not that scared.
No, I'm not that scared. I'm a little scared, but not that scared.
Right. I mean, it's, nobody, or Satoshi Nakamoto is anonymous for a reason. But, yeah.
I don't think I need to go that far because I don't think they see us coming. I honestly think that we'll be on the blockchain and running before they even know it hit them. And governments and big organizations are like Titanics. They steer very slow. Our government can't even figure out what to do about ICOs. Are they good? Are they bad? How are we going to, they're lost. So we'll just keep moving, and we'll just run, and then let them worry about, what are you going to do? You've been left behind. That's the real solution. I don't want to fight anybody.
I think that is what's happening. The faster this goes, the faster this whole technology is moving, and industries are changing because of it. Like, the farther behind they keep getting, because there's too many different things happening at once, and there's no way to actually figure it out.
Totally true. Totally true. Yeah.
Yeah, like, it's really not just Bitcoin and the money aspect of it. It's like there's so many things that are being blown out of the water.
Can I put in an order now, please? Because I would like to remove the gene that gives me sunburn because I'm a redhead.
We'll work on. We'll have that coming soon to a theater near you, I think.
There probably are people who are totally immune to, well, not totally immune to sunburn, but there's definitely people who don't get sunburn as bad as theirs, right? I mean, people in Australia don't wear it all day, do they? So, sunscreen, do they?
Well, my cousin, she's a redhead, and she lives in Australia, and she suffers really badly, and she's got to wear like factor 50 most of the time. Yeah.
Risk wants to be black. Risk, you're never going to be black. Sorry to disappoint.
Yeah, I looked into going the other one. You mentioned the whole sunburn thing. I figured if I were albino, people would take me seriously. That DNA could be cut. I mean, totally full on albino, no pigment in my skin at all. And there's 2 genes I'd have to knock out to do that, to achieve that. I don't know that I'd get red eyes, but that'd be kind of cool too. I don't know.
What if that's what Michael Jackson did?
For now. Really?
Would that be what Michael Jackson did?
You know, I'm not. That's a very good question. It's feasible.
It makes a little sense.
Feasible. I don't. I would imagine that they did that more at because CRISPR really wasn't firmly in science till 2005. So I would expect that they did something to his skin with bleach or a bleaching agent of some sort, rather than actually go in and modify the DNA.
Well, they did.
That'd be my guess.
He did bleach his skin, but that's a good question because the underlying condition apparently that he had is apparently a genetic condition.
Very true. Very true. And there's, you know, we've got solid science on which genes control your skin tone, how much melanin, I'll pronounce it wrong because I read. I don't listen to professors tell me how to do things. I'm self-taught in most of the things I do. So I read a lot, which means I don't hear the pronunciations. So I get it wrong. But yeah, that whole melanin in their body, in your body is, we've got good science on which genes can control that.
Yeah. Yeah. What I heard is that we basically all have the same pigment, but some of us have more of it than others.
Right.
And that's what,
the question just came up in chat about making people docile. I don't know what the docile gene is.
So is there an intelligent gene then?
I would think that there is. There's a genetic makeup. You know, that's just, I don't have a chance to substantiate it with any evidence, but it would be really interesting to have our blockchain with 1 million people on it and figure out, you know, is there? Because we'd know pretty quick. We'd be able to find that out.
do you think there are genetics that cannot work together? Do you ever notice, like, some genetics seem to always come together and others you'll never see together? Well, not genetics. I mean, some like features, some characteristics in people. For example, like certain forms of intelligence, notice that they lack in other areas, like their social intelligence or something like that.
Right. You know, that's really interesting. Again, I think that would be an awesome research question for our data. You know, that is there a relationship between not combing your hair and being super smart? You know, I don't know. I don't know that's, you know, there's a hard thing because I have a pretty spiritual grounding in terms of, you know, what I think we really are. And I don't believe it happens to be this body that is me. I think if you chopped off my finger, you don't have me. If you chopped off my arm, you still don't have me. And if you ground up my body, you didn't grind up me. But I mean, that's just my personal feelings on it. But and I like to believe that I'm in some control of who I am rather than the body tells me everything that I am. At least I'd like to hope that.
Yeah, I get that. I kind of think that way about people who've passed on. You know, you see them, they're still present, so they're gone. You know, whatever trace they left behind, if they've written a book or whatever it is. And nowadays, like we're all leaving traces of ourselves all over the internet.
Absolutely, you steamers.
Yeah. So you're talking about joining Steamer, aren't you? I don't know if you actually signed up for an account.
I just got approved. I got a couple days ago. I got my letter back. I'm good to go. Now I just got to think of something really cool to write. I don't know.
Yeah.
I think you had a topic.
Oh, what could you write about? Yeah.
Yeah, I suppose I'll come up with something, right?
I think you've got a ton of followers waiting to follow you.
You guys are kind. Thank you for that. Thank you for saying so.
Okay, what have we? Is age going to be involved with proteome interactions? What's that mean? I don't know. Or just chiefly focus on genetics and known associations? Proteome, you read that one?
You know, I wouldn't close the door on anything with our data set. We have to see how this evolves. You know, once we've acquired enough different humans' DNA, we could pretty much go anywhere with that. You know, and I wouldn't want to restrict us in what research we would or wouldn't do. I would say that, you know, that's up to the community. One of the things that I'm about is getting out of this once it's established. I'm looking to pull figure out a way that I can get it up and running. establish enough labs so that the flood of DNA can get up onto the chain and properly secured. And once that's in hand, turn it over to the community to say, well, how do you want to mine? That's why we're giving the EOS tokens back to the customer. We're not keeping them for ourselves. We're not going to centralize and control this. We're going to let the community decide what research we're going to get into. You know, what's important? What does the community want to solve? Do they care about cancer? Do they, you know, while we can have a bot looking for patterns, that's important. But again, the community is going to actually have the final say on how it happens. The idea of a bot is just my take on how we could do it a little smarter. But the community should control the bot. I hope that answers your question.
If we only understand 10K or 50K possible, like, genetic problems or whatever, how do you prevent unintended gene mutations that might be even worse?
Really good question. And the answer is CRISPR is extremely accurate. It uses 20-letter pin in order to decide where it's going to edit, where it's going to cut. And that's a long pin, right? 20 digits, except they're only 4 letters, so it's a little less secure than a 20-digit pin, but it knows exactly precisely where to cut. Not only that, you can verify the cuts. So you take DNA, you take a live cell, you CRISPR it, you take the DNA after it's been CRISPRed and grown up, you know, you grow a few of those cells up, and then you run the DNA again, and you validate that you've only cut where you intended to cut. In the beginning, there were precursors to CRISPR-Cas9 that, like CRISPR-Cas3, I think was one where it would cut multiple places and they didn't control it very well. CRISPR-Cas12 just came out, and it's kind of cool because what it does is it cuts, but it also leaves a fluorescent marker where it cut. So CRISPR-Cas12 will be a great tool to double check that my edit's safe, that I've designed the proper edit. When I went onto AdGene's website and I said, here's my 20-letter code where I want you to cut, and we can check it in computers, but it's better to check it in the live cell because biology always doesn't quite behave the way computer bits and bytes do. Did that answer the question? I hope.
Yeah, I mean, pretty much. I've you know, heard kind of anecdotal tales of it not being as effective as some of the, you know, like conflicting news reports, I guess. So I just wanted to clarify whether that was actually the case or not.
When you say there's some efficacy question, is that what we're talking about? How pervasive is the edit going to be? I mean, is it one-for-one edits? No. That's why it's sort of a ongoing treatment, if you will, to slowly over time alter one's genome. But again, it's all. Go ahead.
Sorry. Well, no, what I've read is that CRISPR, and it may have been CRISPR 3 or whatever, didn't cut as precisely as we might want yet. But if there's I mean, it's going to evolve over time and get better anyway.
It sounds like I think you're correct. What you're describing, I know that CRISPR-Cas3 had huge issues. It would cut triple places and sometimes not cut at all. But CRISPR-Cas9, this newer evolution, is damn accurate. It's sweet.
Can I quickly ask as well, the way that you're doing this through the blockchain, how much more quickly do you think you'll achieve the results that you're looking for, as opposed to if you went down the normal conventional route of science?
Yeah, I'll give you some feel for that. When we achieve 100,000 users, that'll be the biggest ability to do genetic research ever, okay? When we hit 1 million users and get 1 million people's, genetic research is over. They're done. You understand? They're like the central banks. They got to go by because there's nothing to do. We have the data. We'll have all the answers. We'll have all 60,000. And how long will it take me to get 1 million users? I don't know. I think at 100,000, we're well on our way to solving most of these problems that are lingered, especially these things that we might suspect the answers are already known. When the guy studying colon cancer runs out and gets 500 of each, that the are the eye color related to the problem? Well, you only see that when your data sample is small. So the answer is, how do you find a needle in a haystack? You add a shit ton more hay, because then all the hay looks like hay and you can throw it away because it doesn't matter. You've got hay on both sides of the sick well.
And Kat probably says with a magnet.
Yeah, sorry about the
I think
swearing,
she might
guys.
be right.
Sorry about that. I like the magnet trick too. When you let me know when you get one. But yeah, that's how it's going to work. With more data, we'll get answers much faster than they're old archaic. I think they're doing it wrong. They're approaching the problem from saying, "Give me some sick guys and some well guys." What you really need is all the data. You know, you need the big pile of data there first, and then you look through that big pile of data and find, oh, anomaly, anomaly, anomaly. Then go find out what those mean and run them back to the diseases, right? You don't start at the diseases and then go pick in the data, because if the data set's big enough,
of other things in there.
all the noise can wash out because it doesn't matter. It doesn't affect somebody's cancer ability if they have blue eyes or green eyes. I hope I said that better. It's kind of a, yeah.
Yeah, I understand.
Dottie gets it.
Cross-sectional studies. What does he mean by that?
That's what I was just kind of describing, that you come from the data and then worry about what the data means after you've found the patterns, as opposed to working it this way, starting at the disease.
It means that if you have a sample of 100 patients, it's not going to give you as accurate a data set as if you have 1 million patients.
Absolutely right.
Yeah.
And that's why I think that today's geneticists, they use words like might be related, is related. They don't say is the cause, and they can't nail it down because their data sets are too small. There's confusion and noise in the data that prevents them. And a guy like Cliff High would totally understand this language in terms of how you got to weed. You find the anomalies first, because that's what he does with the web. He does it with just a different data set.
How do you tell an X chromosome from a Y chromosome?
Is this a joke?
Could be.
I want to hear the punchline, because I don't think I know that one.
You unbutton their genes.
I like that. Can I borrow it? I'm going to put it in the next paper.
Hell yeah, freedom first.
Thanks, brother. That's great. That's a good one.
That's good. Actually, yeah, that's yeah, that's got me thinking now, right? Your X chromosome and your Y chromosome are genes, right? But you can't edit those, can you?
Yeah,
you could, but you wouldn't want to. Well, I wouldn't want to. They're on the germline.
So they say, like, if you have like one extra or whatever, then it leads to what do you call those, like disabilities? So what happens if you have like a whole bunch extra?
There are people that are really sick genetically, and they've got multiple copies of chromosomes all over the place, and yeah, they've got real issues. Most of them don't survive birth. Some do. So, I mean, if you're alive and kicking and reasonably healthy, you can assume that you've got pretty decent DNA. Could it be better? Of course. Could it be worse? Probably. So, I don't know if that actually answers your question. Yeah, there's some really horrible genetic things that can go wrong.
Do you mean, are you asking if you can undo Down syndrome, for example?
No, I was just wondering what happens if you have like a whole bunch of extra chromosomes instead of just one.
I thought you needed
No, you've got 23 times 2, so you've got 64.
Oh yes. Okay.
Yeah.
So it's
all right. So you got 2. That's what you got. And
23 times 2 is 64.
Did I do that wrong? I've been up for a lot of hours, so maybe I need a nap or something. But yeah, okay, I got it. 46. Dyslexia going on. Maybe I'll edit that next week. Take care of that.
Just inject yourself with more of that mathematical gene.
Perfect. I need that one, like in spades. So yeah, you know, that could be horribly wrong. There are people that carry around extra chromosomes and have no impact. So I think it's variable. It depends on how the body, what it does with it. You know, a lot of that stuff, it depends on whether it's encoding or not, whether it's junk DNA or whether it's actually like trying to build proteins and do things, and how clean of a copy is it.
Right.
So yeah, there's a lot of
just
factors there, not just the quantity that it's there. Is it doing stuff, right?
You have an ICO, I think, coming up the 30th of April, or is it before the end of April?
We're going to try to push before. I don't know that it's going to happen. I do know that by the 30th, I want it out and done. I'm focused on that pretty steady right now. So I want to get it off the, we need to get running. We need to get going. And I think we need to do it sooner than later, because waiting around for the government to react to whatever they're going to react to is probably the bad answer. I think we just drive forward, get our, you know, we've got contingency plans where we'll jettison our operations in the States and head to possibly Puerto Rico or possibly even further south, just to get into an environment where we're not going to be messed with. So there are fallback plans, but right now the main one that is in our way is we just need to acquire, the token sale is our effort at we don't want speculators. I'm not very interested in speculation, and I will out-manipulate them maybe. We've had discussions inside our team that if people begin to speculate and drive the price of our token up, we're gonna have to drive the price of our service up. So it's really not a coin you want to buy to speculate with, because that's all you're going to achieve. You know, you may make the token more valuable by speculating on it, but that just drives our service up, because we can't take, you know, the token that's now valued at $1,000 a token that we sold at $100 and give you $800 worth of service, because it's not worth it to us. So yeah, I mean, we'll have to.
I like to hear you say that. I like to hear you say that, because there's so much. A lot of people who are into blockchain, into cryptocurrency, they all have this idea that it's all about money.
Yeah.
You know, it's clearly not about.
To me, to us, to the project, it's about customers. We can do great good if we get enough people in our community, involved with this project, participants getting their DNA onto the chain so that we can see what's what. And our total success depends on how many numbers of people, not how much money we make. The number of people is what's going to give us the power to cure things, to help people, to end suffering. And if we're not focused on acquiring people into the community, then we lose. We're useless. Somebody else will replace us who has a better model.
Yeah, sorry, I'm back. I have some question for you, Simpson, and that's what I was thinking about the environment, because I was thinking that if there's some possibility that our environment can affect in the DNA editing.
Oh, no doubt your environment is constantly affecting your DNA. There's, you know, you go out and get enough sun and you've done damage. As a matter of fact, there's a very interesting article, I'll have to post it back here in this room sometime, of an astronaut that was checked in space with this device on the International Space Station. And he basically ran his DNA while he was in space. He has a twin brother, and he varied from his twin brother quite a bit while he was in space. The article is fascinating. He came back down to Earth, and his DNA went back to normal.
Wow.
Yeah,
that's
that sounds good.
Yeah, so I mean, our DNA is definitely impacted by our environment. There's no question in my mind at all. There's good science on that the environment can change your DNA.
Yeah. So Risk Devin there has the last question. Will it be accessible to everybody or just age?
The data belongs to the community. AGE is a conduit to get the data from a body through this machine onto the blockchain. That's all we're here for. Our only objective is to get people to sign up, give us some spit. We'll turn your spit into numbers, store it on the blockchain where it's safe, and you control who that DNA belongs to. but anyone on the community should be able to access the miner's code to go do research. Does that answer your question, Risk?
Can I sell my DNA?
It's a good question.
Well, that's a really good question, because 23andMe sells your DNA every time you give it to them. If you read the terms of service and terms of use of any of those big guys who are running genetic sequencing right now, you give up all rights to your DNA as part of, just like you do when you sign up for Google. You say, yeah, you can take my data and sell it and
sell
make
my
money.
data, yeah.
And I think that's the problem. I suppose I don't think you should be allowed to sell your DNA, but if you've got a differing argument or a differing perspective, I'd love to hear it. I think that, yeah, go ahead.
What if you could upvote parts of my DNA?
All right, that's cool. I'll have to think about it.
Well, my counter argument to that would be that if it's mine, why can't I sell it? I mean, I wouldn't personally sell it. That sounds insane to me, but if it's mine, I should be allowed to sell it if that's what I choose to do.
If you have a super gene, well, I can imagine why someone would say it's valuable. So, but
yeah,
sorry.
I could see a case for that, and I think my objective is, when I get a community, we'll work out things like this. I don't have a community yet. I've got me and a handful of guys trying to write some code. Once we get involved, my whole objective in giving you guys the customers, the community, the EOS, is that's where the power is. That's what controls the processing of the blockchain. That's what you have to stake for those miners to go do that research and make those decisions. So I'm all about the community taking power away from me. I just need to be here to get this done so that the community can exist. And after that, I'm going to probably sit on a beach somewhere.
I'd say that, like, on the ethical matter, I mean, Steam, for example, right? I've been here since 2016, and Steam went from being completely against vote selling to just, you know what, let's set up a bid bot, and everybody's jumping on that bandwagon. So, like, you don't really know how these things are going to go. Like, ethics change, morals change.
May I ask what blockchain technology it uses for storing your genome info?
EOS.
Sure. We're going with EOS. That has the way they're building that, and the techniques that they have, the tools that they have, it makes a lot of sense for our business. Ethereum just won't cut it. There's just not a blockchain that's going to allow us to do what we want to do. In terms of updates, in terms of linking medical histories with bits of DNA across the chain, EOS is the right platform. So yeah, that's where we're going.
You've been working with those guys. Okay.
Oh yeah, absolutely. Talk to them all the time.
So it uses some kind of delta compression that only stores the differences from a baseline genome?
Correct.
And that genome is kind of like an average of man?
It's a standard in the industry. Is
that like a window doll, like a mannequin version of the human being?
No, it's sort of an average. You were right in the first time. It's sort of an average. It was the most common, not necessarily the true average. So what they did is said, this is the most common condition for the DNA string to be in, and where you vary from that, we call that a delta, and we record where it is and what it is. So that way, you need one copy of the dictionary and your little delta to recreate you.
Would it be possible to produce some kind of an artist's rendition of what that person looks like, that average human?
Wow.
Standard human being.
Yeah, that's a pretty cool question. No, we just don't know enough. Like I said, there's only 1/5 or 1 to 5 of what we do know against what we don't know. So,
can you produce a baby that has that genome?
Yeah, well, that could happen. It would be insanely expensive. Insanely expensive.
Okay. Okay. So, but we can pay for it in Bitcoin.
Not to me. I don't have the printer. You got to go to AdGene to get the 3D printer that prints DNA.
Did we write that down? What's the name?
Thank you, everybody. But thank you so much, Greg, for coming in and talking to us.
Thank you, and thank all of your listeners for the great questions, great conversation. I really enjoyed this, and I think it's helped our project just to be able to vocalize it and get some feedback on how it's being explained. So thanks again for the opportunity to stop in.
Yeah, it's been really exciting. Thank you. So I'm going to switch off the stream. And just so